Howdy, you guys. My name is Mrs. Anitsakis, and I'm going to be your resident scientist for the coming year. Before you guys meet me, I just want to show you guys a little bit about what I do when I'm not going to be in class with you. Behind me is the building, the Oceanography and Meteorology building that I work in. I do all of my lab and other research activities inside this building. And so when I'm not in class with you guys, this is where I'm going to be. But when I am in class with you guys, I want you guys to get ready for all the exciting fun that we're going to be having over the course of the next year. I'm going to be working with you guys one-on-one doing lab activities, introducing you to all the fun that can be had in science and everything that goes on using technology and other lab experiences. So I want you guys to come inside with me for a moment so you can see what I'm doing in the field of science. Here at A&M, we have many different classes. If you come into the Oceanography department, this is going to be one of our major lecture halls. You'll see that there's room in here for over 150 students. So classes here are extremely large. So when you come to college, you might be put into a class like this. But don't be intimidated by the large class size, because in our department we have labs. And so what happens is the students get broken up into groups of about 20 students where they get to work one-on-one with a teacher and another grad student and get to do hands-on labs, much like what we're going to be doing this next year. And that way they can learn what's going on in oceanography. As a grad student, I take 9 hours of credit each year. But since I'm beginning my third year, all 9 hours are going to be doing research. So I actually am done with classes. But most undergrads, I believe, take about 12 to 15 hours of classes each week, which means they're in class for 12 to 15 hours a week, which I believe is a little bit less than what you guys are used to. So that's the good news. Now we're going to go into my lab and see what it is that we do in our lab. The first and most important thing is that when we go into the lab, we always put on a lab coat to protect our clothes. And you always make sure we wear closed-toed shoes so that we don't drop anything on our toes, because we can cut or hurt our feet. So let's go into the lab. So first thing in the lab, we see all these different chemicals and the glassware that we work with. And you'll notice that they're all protected back behind there so that we don't accidentally get any of them on us. So now that we're in the lab, we see that here Shelly, my lab mate, is actually doing some work. Okay, Shelly is loading DNA into the agarose gel. You'll notice that you see a blue liquid. Well, the DNA is stained with the dye so that we can actually see as it floats down the gel. You'll also notice that she's also wearing a lab coat with lab gloves. The stain that she is working with is extremely toxic, and so to protect ourselves from this, we wear gloves and also label the area to let people know not to lean up against it. Another gel that we run DNA on is called the denaturing gradient gel electrophoresis. This piece of equipment, you load the DNA in the top and then it runs down the gel. This takes about 18 hours to run. So usually this is something that I'll load late in the evening, and when I come back to work the next morning, it's ready to go. Once the gel has been running overnight, we'll take it out of the gel and put it into this piece of equipment. Again, before we touch any of this equipment, we make sure that we have gloves on our hands to protect our hands. We can put the gel on this tray and then turn on the UV light. I'm not going to turn on the UV light right now because that's harmful for our eyes, so that's why we keep it in this box. So once we take a picture of our gel on this equipment, we can visualize it using the camera and see it on the computer. What we see here are two different gels. Initially, what we'll first see is we'll see this light banding pattern where the DNA actually glows due to the stain that was put into it, and the UV light makes it glow. So we'll see these light bands, which are the DNA. To better visualize what we're looking at, we'll invert this so that the light bands come out dark, and then we'll rotate it so that the top of the gel is up here and the bottom of the gel is down here. And the DNA is moving down the gel like this. I use this piece of equipment and this gel so that I can visualize the diversity that is in the bacterial community structure. Each band represents a different bacteria that we find in the environment. And so when I have those many different bands, I can see that there are 20 and 30, 40,000 different kinds of bacteria in each environment, and I can compare the different environments to see which bacteria are present. So before I start comparing the DNA of bacteria, first I actually have to isolate the bacteria. Again, using gloves, because I am dealing with bacteria which can make you sick. You can take your water sample and streak it out on plates. And what you guys can see here is we have many different kinds of bacteria that are growing. You can streak it out to where you can get solid plates where it's full of bacteria. And then you can streak it out to where you can get single colonies or dots, where each dot represents a single clone of a bacteria. We can grow these bacteria out on the desk like you see here. Bacteria can also be grown at higher temperatures. To do this, we place them into an incubator. This incubator is like a refrigerator, except for it keeps the temperature inside at a higher temperature, like 60 degrees, like this one's set at. And that way the bacteria can grow at 60 degrees. Here we have a hood. When I turn on the hood, it sends out air that comes through the back of the hood past me. That way, when people are working here, air is constantly being blown out of the hood. Now, the air that is being blown out of the hood is filtered and clean, so therefore anything that we do here cannot be contaminated by air. So a lot of the work that we do with bacteria and with DNA, we do in this hood to prevent contamination, which is a major issue. So now that we've grown up the bacteria, the next thing that I'm going to have to do is extract the DNA from the bacteria. Well, this is done using many different tubes and chemicals and other things like that. So you can see that I have a wide variety of chemicals that I need to use when extracting the DNA. And this is the bench space that I use to do that. Once I get the DNA extracted, I will centrifuge it and collect it into a pellet. This is what we call a centrifuge. Inside, I can place my tubes and spin it down at a high rate. The tubes will be placed in here, and then you seal this, close it, and then this rotor will spin where all of the DNA will be concentrated on the bottom of each tube. Now that I have my DNA extracted, I can continue to do the work that I was talking to you guys about previously with looking at the different bacteria that are present by identifying the DNA. So when we're not in the lab doing lots and lots of work, and when we're not in our office reading papers and working on things, you'll find both my office mate and I, Shelley, and even our advisor in our community area. We come in here to eat lunch, make coffee. Coffee's important. And just sit around and enjoy each other's presence. This is our main meeting room where you'll see most of us get around and just chat throughout the day and talk about our projects and try to problem solve things. You'll notice though that in our community area we do have this one piece of equipment that we work with. This is a special hood. In this hood we work with chemicals and primers and other things along those lines, and under no circumstances do we allow any DNA to go in here. That way all of our chemicals that we work with never become contaminated with our DNA. So another important aspect to what we do is actually storing our samples and storing our water so that we can look at them later in time. What we have here is a walk-in fridge. You can walk in and feel that it actually gets pretty cold in here. You can come over. In our walk-in fridge, we also have lab space so that we can do work in here, but still maintain the temperature at 4 degrees Celsius. What we see here is we're forming those bacterial aggregates that I was showing you guys on the microscope. By rotating the water, all of the particles in it slowly congregate together, and they form aggregates. And you can even see them in the water. Okay. This is a process that takes a lot of time, slowly rolling them. These have been on here for about 3 or 4 months for them to get to that process. the water that you see sitting in there was actually collected from these, what we call mesocosms. What we do is we take seawater and we let it grow and change based on different things that we want to look at. Like say we'll add nutrients to it. And that way we can experiment with seawater and experiment with the environment, but do it in the safety of our own lab. On top of our walk-in fridge, we also have a walk-in freezer. And this is kept in a minus 20 degrees Celsius. Do you guys know how cold it is? I'll find out when I come see you guys. But this is our freezer. It's a minus 20. You have to wear a coat if you're going to work in there. So one of the most important things that you guys will find out about science is that after you do all of this hard work in the lab and research, you want to be able to show everybody what you have discovered. So what we do is we will put our information together and we can do publications. Publications, you can see, are journal articles, much like those that I read to get information. We will put our information into an article and publish it. Another, a little bit more informal method of giving our information out to the scientific community is by creating posters. Posters are kind of like journal articles, except for it's a little bit more based on just information that you've discovered. It's a little less wordy and has more pictures and diagrams. In our lab, we've put together many posters and presentations at different conferences. Just last year, I was able to go to Salt Lake City for a week and present my research, which is out here in the hallway because it's the most important, so we want people to see it when they first come. So you'll see again some of the same things that I was showing you guys in the lab. Is I put together pictures and graphs of the stuff that I have been working on, so I can go show it to other people around the world. What you guys will see here is this is a picture of a filter. This is the filter that I collect my bacteria on. I force the water through this, and all the bacteria stick to this filter, and then I can pull the bacteria off that filter to get the DNA and other information from it. Another important aspect of what we do in microbiology is we also have to quantify how many organisms are in the water. So some of the things we do, as my advisor here, Dr. Bonk, is doing, is he's counting bacteria on the microscope. If you zoom in on the screen, you can see all of the little bacteria on there, and I'll let him explain exactly what he's doing.
So to visualize or count the bacteria, we first have to stain them with a dye so that we can see them under the microscope. And so what we have here are different organisms. The blue ones represent bacteria. That's because they are stained with a dye which binds to their DNA. And then we also see red dots here which represent other bacteria but that are capable of photosynthesizing, which Erin will explain to you one day, hopefully. And so they have certain pigments that absorb light, and then they emit light at a different wavelength, and hence they have this red color. So we can distinguish bacteria from photosynthetic bacteria.
And sometimes we see that bacteria are evenly distributed across the field, but other times bacteria can aggregate together and form clumps, much like what we see in this slide. So when Shelley and I aren't in the lab, we come sit in our office and research using papers. Usually my desk is much more disorganized than this. You'll see my desk covered with all these journal articles where I read and get information about what other people are doing on their research. But when I'm not wanting to think about research, I can come in here and look at pictures of my friends and family. I'm originally from Dallas, Texas, and I went to high school in there. I believe some of you already know that my graduating class had over 800 students in it, much larger than what you guys are used to. So I love to have pictures around me that remind me of all my friends and family back home in Dallas. Also on my walls, I have certifications that I've received that allow me to work in the laboratory. And on top of those, I have newspaper articles that have been published about my research. Newspaper is another way that scientists relay their information to the public instead of through journal articles. So one of the first things you might notice when you come into my office is this very, very large map of the Gulf of Mexico. Well, this is where most of us do our research in the Department of Oceanography. You see in this red box, that's my study site. This is the west of the Mississippi River in New Orleans along the Texas-Louisiana shelf. And I collect my water samples from this area. Behind me, you guys might notice all the folders and books that are on the shelves. Well, these are from all of my classes here at A&M. Just my classes here at A&M. Right now, I'm starting my 3rd year of graduate school, which to you guys might be the equivalent of my 19th year of school. So all in all, I've spent a really, really good time in school. But that's because, you know what, school is fun, and you guys will enjoy it too. The one thing that I really, really like about graduate school is a lot different than high school and middle school and just regular college on top of that. In graduate school, I have the ability to choose what classes I want to take. All of these books that you see up here are for classes that I really, really wanted to take because, you know what, they're enjoyable. And on top of that, I get to do research in an area that is exciting to me. So that's what I love most about college, is that I get to choose what I study. And my advisor, who you met earlier, he's there to kind of help me when I come along problems and to help me think about other things that would benefit me if I were to take that class. So one of the most important things about going to school is choosing something that you enjoy. I enjoy oceanography. I've always enjoyed oceanography, and what got me interested into oceanography is scuba diving. I have been scuba diving for many years now. And what you guys can see is my background on my computer is a picture that I took when I was scuba diving off of Cozumel. This is a sea anemone, and you can actually see the little fish that are living within the sea anemone. And just being able to visit these environments and to take pictures of them and to enjoy them really got me interested in oceanography. Another benefit of oceanography is that I get to travel. As you guys have seen on my map, I've been able to go all throughout the Gulf of Mexico. While other researchers are doing studies in Antarctica, in the North Ocean, in the Pacific Ocean, in the Atlantic Ocean, in all of the oceans and seas around the world. So within oceanography, I'll have the ability to travel the world and visit different sites so that I can do research. So if you guys ever come visit us in our lab, one of the first things that you might see is this large display case. In the display case are preserved organisms found from all over the ocean. We have fish, seashells, crabs, and many other different organisms. It's just amazing to see the different kinds of things that you can find inside the ocean. So when you guys come visit, I want you guys to be able to pick out one item that you found on this and show it to me later on.